is Diagnosis Glaucoma with your hosts, Dr. Mona Kaleem
and Dr. Harry Quigley.
Thank you all for joining us today. Harry is off, but we have a very special guest on our program who's going to talk about stem cell therapy. Please help me welcome Dr. Tom Johnson, who is my colleague at the Wilmer Eye Institute. Tom, can you tell the listeners a little bit about yourself?
Sure. Well, thanks very much for having me today. So as you mentioned, I'm an assistant professor of ophthalmology here at the Wilmer Eye Institute at Johns Hopkins. And like yourself, I'm a glaucoma specialist. But I'm also a neuroscientist. So I've been here at Hopkins for a while. I went to medical school and residency and did a glaucoma fellowship here. But before that, I did a PhD in neuroscience at the University of Cambridge in the UK. And that's how I got interested in stem cells in the first place.
And can you tell all of us what made you go into glaucoma?
Well, I think it fits well with my interests in neuroscience and the science of how vision works. I was interested from even high school on in understanding how the nerve cells inside the eye and the brain process vision and how electrical impulses in those cells actually translate into pictures in our mind that allows us to see. And when I did my PhD, I worked on degeneration of the optic nerve. Glaucoma is the most common one, and we'll talk about that today. But I was interested in why nerve cells die in the optic nerve and what we can do to protect them or regenerate them.
Tom, I'm really impressed that you also got a PhD. Can you tell us a little bit about what you did your PhD work in?
Sure. I think it's actually a good story because it gives some insight into why what we're doing today is so exciting. So I was interested in stem cells because this was something that was new and exciting in 2005. And this was actually a time in the United States when there was a lot of debate about the ethics of human embryonic stem cell research and what could be done with federal funding with stem cells. And because of the uncertainty over federal funding for this kind of research, that's why I actually went to the UK. I did my PhD with Keith Martin, who incidentally was a fellow here with Harry Quigley just a few years before that. We were studying whether there was a role for stem cell transplantation as a therapy for glaucoma. Now, at that time, I would say the field was not far enough along to be able to regenerate the optic nerve. We didn't know how to take stem cells and turn them into the types of cells that live in the optic nerve and how to make them conduct vision. And so instead, I focused on using stem cells to provide support and therapeutics to the optic nerve in models of glaucoma. So in essence, how to provide neuroprotection and how to slow down the nerve from dying in models of glaucoma.
You bring up an important point. This is kind of a bit of a controversial topic. Some of the work in the United States has, would you call it, maybe lagging behind what we're doing in some other countries?
That's what I was afraid of at the time, and I would say that may have been true for a certain number of years. But the field of stem cell research was transformed actually in the mid to late 2000s with the discovery of something called induced pluripotent stem cells. It was actually a discovery that was awarded the Nobel Prize just a few years ago. From the 90s until the mid 2000s, the only way to obtain pluripotent stem cells, meaning stem cells that were capable of forming any cell type in the human body, was to create embryonic stem cells. So induced pluripotent stem cells are a different technology when you can take any cell in an adult human, like a skin sample or a blood sample, even a urine sample, and you could take cells from that sample and in a dish manipulate them so that they turn into stem cells. And so we can create then stem cells from anyone that we want to treat and potentially give them back their own cells after we've manipulated them in a petri dish.
Oh, wow. That's really fascinating. And I wanted to ask you a little bit about the optic nerve and glaucoma. We do actually have an episode for all the listeners. It's episode number 8 on the optic nerve. But Tom, can you kind of just lay it out for us with all of your expertise? What happens to the optic nerve in glaucoma?
Sure. So I think the thing to understand about optic nerve disease in general and why the optic nerve is so critical is you can think of the optic nerve sort of like the cable that connects your cable box to your TV. If you damage that cable or you cut it, even though the cable box works and the TV works, there's no picture coming through the TV. So the optic nerve is just like that. The retina inside the eye is like the film in a camera. It takes light and turns it into an electrical signal, but it's the job of the optic nerve to take that electrical information and convey it to the brain. And your brain needs to have that information in order for you to have vision. So glaucoma is an optic nerve disease. It's the most common optic nerve disease, but it's not the only one. And all of these optic nerve diseases lead to degeneration of this cable. And the problem is in human beings, and in fact in all mammals, once the cells that comprise the optic nerve degenerate or die, they're not replaced or regenerated. And so optic nerve damage and the vision loss that goes along with it is permanent.
So can we stop or prevent the loss of the cells?
Yeah, it depends on what disease you're talking about and why it's happening in the first place. We, on a day-to-day basis, deal with glaucoma, and I think most of the patients listening to this podcast are interested in glaucoma. So the good news there is we do have lots of good ways of slowing down and in most cases stopping the damage from getting any worse. Interestingly, it's all based on, right now, lowering the intraocular pressure, the pressure inside the eye. That's the most important risk factor for glaucoma. It's the main driving force for the death of retinal ganglion cells, or RGCs. Those are the cells whose fibers comprise the optic nerve. So when we talk about optic nerve cells, we're talking about RGCs. And as those die from high pressure, that's permanent. But if we can get the pressure down in patients' eyes, then the optic nerve damage slows down or does not get any worse.
So in addition to stem cell therapy, will patients still need to use eye drops to lower their eye pressure?
Yeah, I think that's critical to understand because, high eye pressure is damaging to the optic nerve cells that a patient was born with. Then there's every reason to believe that even if we can put back new optic nerve cells with stem cell transplantation, those are still going to be susceptible to high pressure. So I think controlling the pressure is always going to be one of the most fundamental things we need to do for patients that have glaucoma.
Okay, great. So this is kind of like a complementary factor to lowering the eye pressure with medications and surgeries.
Yeah, exactly. I think forever glaucoma, the number one thing will be slow down, stabilize the disease, prevent it from getting any worse by getting the pressure to a lower level. But if we're successful in developing stem cell therapy, we could do something that doesn't exist right now, and that is potentially actually bring back vision that has already been lost.
That's really amazing. We get asked tons of questions on that very topic. What we're doing in terms of stem cell therapy, if there's some kind of way to regenerate the nerve tissue. How far of a plan do you see this as? Because we get asked about that all the time. Like, what is kind of like the timeline for this work?
Yeah, at this point, it's almost too early to realistically speculate on that, I have to say. Right now, the work we're doing in the laboratory is focused on stem cells in a petri dish and in animal models like lab mice and lab rats. We are working very hard to figure out the limitations and the barriers to getting this to work well in those animal models. And as we figure that out, we'll be able to move this forward into other models and eventually into humans. But realistically, at this point, we're thinking that it's probably going to be on the order of at least 5 to 10 years before we're ready to think about doing this in people, because safety has to be the number one concern.
Now, in glaucoma, what we usually tell people is that the vision that you lose cannot be regained. So are you in fact saying that we can bring back vision that's lost, or we can just try to slow worsening?
So You mean with stem cell
With stem cell,
transplantation?
yeah.
Well, the ultimate goal is to bring back vision that's been lost. So when I think about how this is most likely to play out, I think the first patients that are going to benefit from stem cell transplantation are going to be people that have absolutely no vision. People that have what we call no light perception, where they can be in a dark room and have the brightest light you can imagine shown in their eyes, and they really can't tell if it's light or dark or not. And if we're able to take stem cells and regenerate some of those optic nerve cells for them, then they may get some rudimentary vision and be able to see light versus dark, see shapes or movements. And then ideally, what we hope is that as we gain more knowledge and have more nuanced approaches to regenerating these connections between the eye and the brain, we might be able to get better visual outcomes.
So then patients could potentially go from seeing no light to being able to see maybe like a hand or some fingers in front of their face and some objects being waved in front of their eyes. Is that correct?
That's our thought, and there's early work in preclinical models, like animal models of optic nerve disease and optic nerve regeneration, that suggests that some rudimentary regaining of vision will probably be possible, but it remains to be seen whether that will happen for humans or not.
All right, we're going to get a little bit scientific here, and I want to ask you a little bit more about a term you just mentioned. You had mentioned RGCs, which stands for retinal ganglion cell. Can you just kind of define that a little bit better in just like layman's term for everyone to understand?
Yeah, absolutely. And I think it's important so that if you understand what an RGC is, you can understand why it's such a complex problem to be able to replace an RGC in the eye and the brain. So an RGC is a nerve cell. Nerve cells basically are cells that are specialized in carrying electrical impulses. So the brain is composed of billions and billions of nerve cells, and the spinal cord is composed of nerve cells. And the back of the eye contains a structure that I talked about previously called the retina, which is also composed of nerve cells. So the retina is sort of like brain tissue in the back of the eye. And it's specialized in that it has a type of cell called photoreceptors that when light hits them, they convert light energy into an electrical impulse. And they send that electrical impulse to a couple of other types of neurons in the retina. And eventually, it gets to the final cell type in the retina, which is the RGC. So the RGC, part of its job is to listen to all of the other nerve cells in the retina and take all of the visual information together, process it, and collate it into a usable visual signal. And then it communicates that to the brain. So the retinal ganglion cell is a very interesting type of cell because it has processes in the retina that allow it to listen to all of the other neurons. And then it has one very long fiber that's called an axon that travels all the way from the eyeball through the optic nerve and to various structures in the middle of the brain. It's a matter of several centimeters in people. So it's a single process that travels a very long distance to send visual information to the brain.
And so the retinal ganglion cells come together to form the optic nerve.
That's right. The optic nerve really is a fiber optic cable where all of those fibers are the axons of the hundreds of thousands or maybe a couple million retinal ganglion cells, along with some blood vessels and some other support cells called glia.
Okay, so let's move on to an actual stem cell. So what is a stem cell?
I would say that a stem cell has two main characteristics. Number one is that it's capable of infinite self-renewal, meaning that it can divide in half. You can have one stem cell and then it will divide and make two stem cells, and two stem cells will make four, and four will make eight, and so on and so on. So if you start with one stem cell in a dish, you can, in theory, create billions and billions and billions of stem cells and have enough to regenerate all of the optic nerve neurons for millions and millions of people. The other characteristic is that stem cells are undifferentiated, meaning they're a very generalized type of cell that on their own don't exhibit any unique features of the tissues in our body, but are capable of gaining those features over time. And that process is called differentiation. You could start with a stem cell that is capable of turning into an optic nerve neuron, or a heart muscle cell, or a skin cell, or an intestinal cell. And so when it has the ability to become anything, it's called pluripotent. And when it actually differentiates and becomes those things, that's called differentiation.
And just out of curiosity, are we doing stem cell work with any other medical conditions outside of the eye that you know of?
Yeah. This is a field called regenerative medicine, and this has been a hot topic for a couple of decades now. Really, you could think of pretty much any medical condition and think of probably several ways to apply stem cells to it. Anywhere where a certain type of cell in the human body becomes diseased or degenerates or dies could potentially be regenerated with stem cells.
Now, there are potentially some ethical and logistical type of issues behind stem cells. Can you just touch on that, please?
Yeah, so the human embryonic stem cells that I alluded to earlier on have been grown since the 1990s, but in order to obtain them, you have to start with a human embryo, and usually those were obtained from embryos that were created for in vitro fertilization and then were not going to be implanted to turn into human babies, and so they were used for research. And so based on those, there are dozens and dozens of human embryonic stem cell lines in existence. And because these are capable of self-renewal, like I was saying earlier, these same stem cell lines have been grown in cultures all over the world for decades now, and we can still use them today without having to sacrifice or use further embryos. But the idea of creating new stem cell lines through the use of human embryos is potentially an ethical issue for discussion. And so that's why using these induced pluripotent stem cells, where I could just take a skin sample from a patient in the clinic and create a stem cell line from them, that's one of the reasons that's so attractive.
So moving beyond some of the ethical issues, if we can make a retinal ganglion cell from stem cells, then what's the barrier to transplanting them in the clinic now?
Well, that's a good question. I want to touch on that point before moving on too quickly, actually, because that was a huge sticking point for years, is being able to make retinal ganglion cells from stem cells in a dish. When I talked about starting my PhD in the mid 2000s, that was something that no one knew how to do. We had embryonic stem cells and we were about to get induced pluripotent stem cells, but how to actually change the gene expression pattern in those cells to make them optic nerve neurons was completely unknown. And that was something that was finally only figured out about 5 years ago. One of the labs among the several worldwide that did that was Don Zacks here at the Wilmer Eye Institute, developed not only specialized reporter lines of stem cells, but also the concoction of soluble factors and ingredients that you needed to put in the culture media to create retinal ganglion cells in a dish. So that in and of itself, I would say it was a huge discovery. But you're right. So now that we have ganglion cells in a dish, the real question is, how do we get those into the eye? And I would say there's probably a number of ways that we could just put them into an eye. That's almost the easy part. The hard part is getting them to actually make the right connections inside the eye and the brain so that they begin communicating with the retina. Because at the end of the day, as I was trying to say earlier, I think RGCs are interesting cell types because they're sort of like communication stations. They listen to the other neurons in the retina and then they convey that information to the brain. And so they need to make connections with neurons in both places in order to do their jobs correctly.
And how will you actually get the RGCs and the stem cells into the eye? Is it like an eye drop or a surgery or what?
Well, I think most likely it's going to be a surgery. We're doing experiments where we try the least invasive way we can think to do this is injecting them with a needle. And that sounds very scary, but in fact, patients with macular degeneration and diabetic retinopathy may know that it's now trivial for people to get injections into the eyeball on a monthly basis for years at a time. So that's something that is not very risky, and we do it relatively routinely. But I think in order to get the stem cells into the right part of the retina and get them into the right layers with the factors that will help them make the correct communication within the retina, it's probably going to be a surgery and a little bit more nuanced than just injecting them into the eye.
This is probably a little bit too early for you to comment on, but what do you think would be like the recovery period?
Well, I'll tell you, the recovery period as far as when will people be back to their baseline, back to, where they were before the surgery, maybe a couple of weeks. If I think this is going to be something that we elicit our vitreoretinal surgery colleagues for, because I think we're probably going to have to take the jelly out of the middle of the eye with something called a vitrectomy. And then a lot of times they'll want to put a gas bubble in the eye that goes away but requires some special positioning for the patient while the gas is being reabsorbed, and that can take a couple of weeks. But I think it's important to note that I wouldn't necessarily expect the stem cells to start working for probably months after they were transplanted, because we're going to put them in the eye and they're going to have to grow an axon several centimeters to get all the way to the brain. And there's no way that scientifically you would think a patient would regain any vision until those brain connections were actually established.
What are some of the safety factors behind all of this?
I think this is one of the major concerns we have. This has the potential to be a revolutionary therapy, but at the same time, there are several points of concern about putting stem cells into the eye that we need to hash out before we try this in human people. So one of the main concerns is that I've already told you stem cells have the capacity for infinite self-renewal. They can divide and divide and divide. And that may make you think about another type of cell that sometimes comes up in medical podcasts, and that's cancer cells. So we want stem cells to divide when we have them in a dish and we're creating RGCs. But once we get them into the eye, we don't want them to divide anymore. We don't want them to create tumors. We're going to need to work out the logistics of the surgical transplantation technique. I would say anytime an eye has surgery on it, there are risks like infection and bleeding that need to be minimized. And then I think we need to be able to predict what these cells are going to do after they're injected into the eye. This is one point that I think it's very important for all the listeners to understand, is that at this point, we don't have enough information to be able to predict the behavior of cells after they're transplanted into the eye. And so I don't think at this point it's safe for patients to be thinking about having this done outside of a well-regulated clinical trial. And I just want to bring up the example of an article that was published in the New England Journal of Medicine in 2017 that detailed 3 patients that had a type of bone marrow derived stem cell injected into the eye for the treatment of macular degeneration. And it was kind of unclear whether this was truly a clinical trial. I don't think it was associated necessarily with a university. But those patients all ended up having very severe complications and blindness as a result of the fact that these stem cells created scar tissue in the eye and caused retinal detachments. So, I think clinical research is important, and having patients participate as subjects in clinical trials is important. But that should always be done in the context of a reputable university and what's called an institutional review board and a safety board that has oversight over what's going on.
And where are we right now in the process here in the US bringing stem cells for the treatment of glaucoma?
For glaucoma, we're pretty far from clinical trials. I would say as far as regenerating optic nerve neurons, like I said, we're probably 5 to 10 years off. There are potentially applications of stem cells not to regenerate the nerve, but to offer protection to the nerve by secreting factors that are sort of like medications to prevent optic nerve degeneration. And some of those may be much closer to clinical trial. And, to give some optimism to the listeners, there are other eye diseases where we're actually much further along, partly because the cell types that are under investigation to be replaced are simpler than RGCs. But I think it's giving proof of concept that stem cell transplantation for eye disease is feasible. So in diseases like macular degeneration, retinal pigment epithelial cells are being transplanted. Those are derived from stem cells. And I think in the near future, photoreceptors like rods and cones are probably going to be transplanted into human patients as well. So optic nerve disease is lagging behind a little bit because it's a much more complex and difficult problem, but I do think we'll get there eventually.
And Tom, what are your projects? What are you doing in terms of stem cell therapy?
So my goals are to figure out how we can get transplanted stem cells to integrate into the retina. So again, if we talk about the two-way communication that RGCs engage in, they talk to the brain, and there are a lot of labs all over the world that are doing really great work and trying to figure out how to get ganglion cells to grow long axons into the brain. But fewer labs are understanding of how to get retinal ganglion cells to grow into the retina. And so that's what we're looking at. Recently, we have found one particular barrier that is an anatomical structure in the retina that seems to act almost like a fence to keep transplanted cells out. And under normal circumstances, that may be a good thing, but when we're trying to get stem cells to communicate with the retina, it seems to be a real barrier. We've developed methods, at least in animal models, to circumvent that barrier. And so far, we've been able to get a lot better integration of transplanted RGCs with a recipient retina.
Well, I'm going to speak for Harry and myself when we say that your work is really exciting. We're really proud of everything that you're doing, and we're so happy that you could join us for this episode today. There are listeners who want to know more about what you're doing and support your work. How can they do that?
Thanks very much. It's really great to be here, and I think this is an important topic, so I'm happy to share the knowledge and the news with your listeners. I would say what we're doing is cataloged on the Wilmer website. We're in the process of building a website specifically for the optic nerve regeneration initiative that we have here at Wilmer. I'm one of the directors along with Don Zak, and then my personal laboratory has this and a few other projects going on as well, and the details of that can all be found on the Wilmer website.
If anyone is listening who wants to support or know more about Dr. Johnson's work, you can actually go to the Glaucoma Center of Excellence website from the Johns Hopkins Institute main page. And on the Glaucoma Center of Excellence website, you'll find a link to the development office where you can find out ways to support Dr. Johnson, and specifically you can reach out to Megan Leffner. All right, so Tom, I have been hearing a lot about something called the Argus 2. Does that have any role in glaucoma? I've heard that takes people from seeing no light to then being able to see light again. Is there any role for that in glaucoma, and what is that for? What do you think about it?
Yeah, it's a great question. So, it's a really interesting piece of technology. The Argus 2 is an implantable device that is an electrode array that is actually meant to stimulate the retina directly. And it's not meant for glaucoma. It's a little bit different. If we go back to what I was talking about before about how the retina works, there's a type of cell called the photoreceptor. Also, there are two types of photoreceptors, rods and cones, and their job is to sense light and turn that into an electrical impulse. And eventually those impulses get to the retinal ganglion cells and the optic nerve. So there's a different class of diseases where rods and cones die. And one of those, for instance, is retinitis pigmentosa. And the Argus 2 is aimed at patients that have those sorts of diseases where they don't have photoreceptors anymore, but they still have working retinal ganglion cells and a working optic nerve. So the idea behind the Argus 2 is that a patient would have an electrode array implanted into the eye that is capable of creating electrical impulses that stimulate retinal ganglion cells directly. So they sort of substitute for the photoreceptors. They bypass the photoreceptors. And then a patient has to wear a camera that's usually mounted onto like a pair of sunglasses, and that is then connected to the electrode array. So the camera then picks up what the patient should be seeing and encodes that information into the electrode array, and patients then are able to see things. Now, in practicality, I think the amount of vision that people get from that is not quite as robust as you would think hearing about that. But like you said, people can go from not seeing anything at all to seeing light or maybe some rudimentary motion. But I think as the electrode arrays get smaller and more dense and the technology increases over time, I think that will probably improve. But I do want to tell you briefly about some other interesting technology. There's another device under development that I've heard about. I think it's called the Orion, that works in a very similar way in bypassing the type of cell that is lost that would actually potentially be applicable to patients with glaucoma. So if you think that in glaucoma the problem is that the optic nerve is not functioning properly, then you could bypass the optic nerve by going straight to the brain and stimulating the brain directly. And now the optic nerve projects to a part of the brain that's very deep in the midbrain called the thalamus. It's very difficult to get to from a neurosurgical perspective, but eventually those impulses make their way to the very back of the brain, to a part called the occipital lobe. And so the idea behind this device is you could implant an electrode array into or near the occipital lobe and stimulate the brain directly in a way that would recapitulate vision. I think we have a lot to learn about the neuroscience behind exactly what pattern and how you need to stimulate the brain or the retina in order to get really good vision. But that device, in the few people that have received it under experimental conditions, have again gone from seeing absolutely nothing to seeing sparkles or, little lights that do correspond to the actual environment and help them, do things like navigate.
For those of you listening, you might think that going from seeing no light to some light is really not a big deal. But it's actually a profound change for someone who cannot see anything, whose life is totally in dark. So being able to see a sparkle or a glint of light or a shadow or anything like that is really just tremendous. All right, Tom. So one last thing that I wanted to talk to you about. We hear all of these ads and see things online, and people always talk about lifestyle modifications or dietary supplements or vitamins that you can take to regenerate your RGCs or your optic nerve. Have you read anything like that, or what's your opinion? And share whatever you know with us here.
Yeah, there are lots of such claims. I would say that there's very little scientific evidence to back up most of that stuff. If we want to talk about lifestyle modifications, I would say that there's actually some decent evidence that aerobic exercise probably does help neural function in general and may help retinal or optic nerve function. And so I think that's a thing that I'm comfortable recommending because it may help patients with glaucoma as far as vision, but I think that the benefits of exercise and a healthy lifestyle are robust for the entirety of someone's health. So I think that's good advice no matter what. As far as nutritional supplements, I'm not aware of anything that has actual good scientific data behind it. There are a couple of supplements that, when given to mice, in huge doses do protect the optic nerve from degeneration in models of glaucoma, but the doses that those mice got were equivalent to humans having to eat kilograms of pills a day. It's just something that's not totally feasible. So I think that sort of research is more in the line of trying to target the pathways that those drugs and supplements are acting on and make more powerful medications that target the same pathways.
All right, so for all the listeners, these are your take-home messages. There is stem cell therapy in the horizon. Be patient, it's coming, and Dr. Tom Johnson is one of the people who is bringing that to us. Beware of any fraudulent claims about the RGCs and optic nerve and things that can regenerate it. Try to listen to the evidence-based medicine, so things that have data to support them. And continue to use your eye drops. Don't forget about the use of eye drops, surgeries, and lasers to treat your glaucoma. Again, thank you so much to Tom for joining us today. If you'd like to know more about his work, there are several ways to find out. First of all, go to the Glaucoma Center of Excellence website on the Johns Hopkins Wilmer Eye Institute page. You'll find his profile, and you can reach out to his development officer, who is Megan Leffner. You can also go to our website, www.diagnosisglaucoma.com. You can find a link to all of that from our site. And you can also email us, and our email address is diagnosisglaucoma@gmail.com. Thanks for joining us. Until next time.
Thank you for joining us. Until next time, your mom says take your drops.